Hello students, in today's class we are going to learn about qRT-PCR, which stands for Quantitative Real-Time PCR. So quantitative real-time PCR enables reliable detection and measurement of products generated during each cycle of a PCR process. This technique involves use of an oligonucleotide probe, which is designed to hybridize within the target sequence. Cleavage of the probe during PCR because of the 5' nuclease activity of Taq polymerase can be used to detect amplification of the target specific product. So coming to the principle of quantitative real-time PCR. So this is basically a PCR-based amplification method in which we make use of a probe molecule. So here we have a probe molecule that is complementary or specific towards a particular target. To the probe is attached a reporter molecule and a quencher molecule. When light radiation falls on the probe, the reporter molecule will emit fluorescence, and the fluorescence is absorbed by the quencher molecule, which is also present on the probe. Now during amplification, so here we have the target that the probe that will bind to the target DNA segment. And during amplification, because of the activity of Taq polymerase, the probe will get degraded. Now after degradation, again when the light will be incident on the reaction, the reporter molecule will emit fluorescence. However, the quencher molecule will not be at an optimal distance because of the degradation of the probe, and hence the fluorescence will be recorded by the instrument. So here you can see how the changes take place during each PCR cycle. So here we have the first PCR cycle. We can see the probe is attaching to the target, and then we have all the other ingredients like the primers and DNTPs buffer and the enzyme that is required for the amplification process. So the Taq polymerase will amplify, and in the process, the probe that is already bound to portion of the target DNA is being degraded, and this results in separation of the reporter and the quencher molecule. So at the end of the process, when we have light radiations incident on the reaction, the reporter will emit fluorescent light, which is detected by the machine as the quencher molecule is not able to absorb the emitted fluorescent light. Now here you can see if we have one DNA molecule as the starting material, then at the end of the process, you're going to have 2 DNA molecules. So now in the second cycle, 2 probes can bind. So in this way, at the end of each cycle, the signal molecule is going to keep on increasing because more and more probe molecules can bind. As a result, more and more probe degradation and hence increase in the recorded signal. Similarly, here with the help of an amplification plot, we can see that if we compare these 2 plots, the one in the blue and one in the red, we can see that as at each cycle, we are basically recording the fluorescence, and at regular time intervals, we can see the signal keeps on increasing. So we have basically a plot of your cycle number versus the fluorescence that is emitted, that is recorded on the axis. So suppose if we are comparing these 2 lines, we can see that to reach a signal of about 1000 fluorescent units, the cycle number that is required by this blue line sample is about 19 cycles. While if we compare it with the red line, to reach the same signal, it requires about approximately 24 to 25 cycles. So why this difference? The difference is because in the starting cell number. So if the starting cell number is high, then a particular level of fluorescence is reached in less cycle numbers, while if the starting number is less, then to reach that particular fluorescence signal, more cycle numbers are going to be required by that sample. So on this basis, we can basically quantify how much starting material of a unknown sample is present in the given sample that we are analyzing. So for this, we first need to plot or have a standard graph of known concentrations of cell numbers and what kind of plot we get with that. And we once we have our unknown sample, we can compare it with the standard graph, and we can make a analysis on what is the amount of starting material. So the starting material can be any type of, say, infectious particles such as RNA infectious particle or DNA infectious particle. If we are using a DNA infectious particle, then we can directly quantitate it. If we are using RNA infectious particles, then we need an initial step of reverse transcription step to convert the RNA into DNA, and then we can perform the quantification reaction by using the real-time PCR step. So coming to the last point, that is applications of real-time quantitative PCR. So it is basically used for relative and absolute quantification of gene expressions. It can be used for validation of DNA microarray results, and it can be also used for counting bacterial, viral, or fungal loads in a given sample. So that's it about real-time, quantitative real-time PCR. Thank you very much, and kindly like, share, and subscribe if you like the content.